I'm one of those researchers that started 12 years ago doing still a lot of animal work, research, and we switched actually quite a lot, moved away from animal work and going to stem cell work. And by doing this, when we are asked to debate on this topic of animal use, what you often see is that people like to put you in a corner, and you're like a stem cell researcher or you are animal researcher. And I think I never feel comfortable with that, because what I would like to show you today, that there are very clear reasons why we do research with stem cells or with animal models. And I think you did nicely illustrated that by saying that actually the scientific question is actually prevailing. And I just want to give you some examples of that. And along the line, along this talk, you will also see that indeed every model has its pros and cons. And then the last part of my talk, I will actually leave the word to a group of people actually we don't hear so much about. It's actually the parents and the patient organizations who often support our work and have to deal with the disorders we are investigating. And in my opinion, we don't hear enough from them, like what they actually think about use of animals or stem cell models. So, well, let's start. The very big question that we have, and I think many of us, is actually really try to understand how we can go all the way from DNA to phenotype in terms of behavior. How does it work? How do we how are genes actually coding for our behavior? And the question that we ask is, the reason we ask is that we know that many of disorders are actually caused by mutations or errors in this DNA, and that leads to altered phenotype or behavior. And so the translational cycle that we have always, or approach that we always have taken for many years is actually starting with the patients that have a certain brain disorder. And here at the Radboud and department where I work, those are typically neurodevelopmental disorders. Think about autism, intellectual disability, or epilepsy. And what we are very good at is actually from those patients, really find where it goes wrong, right? Where is the mutation happening in our genes, so we can identify those genes. And then once we know this, we can then try to model it, try to understand the disorder, and that's on this part of the graph. And so by trying to understand what's happening, we can try to and that, for example, we can try to then use those mazes to try to understand why a genetic change actually leads to another phenotype. And then once we have understood this, we can try to manipulate it and hopefully come to treatment strategies. Now this approach is I think very valuable, and we have learned a lot by doing this. But there are a few disadvantages by doing this. First of all, we need this a priori knowledge. So we need actually to know exactly what the mutation is. We need to be able to also to mimic it in the animal models. And I know we often say that the genome between animals and even fruit flies and humans is 99% identical. But that's of course kind of boutade in a way, because now, I think a month ago, we sequenced actually the whole genome, and we know that a lot of parts of the genome that we thought was not coding for any gene is actually very important, and that's actually very different between humans and animals. We also know that the human has a very specific development pattern and physiology, and I will come back to that. And also that the genetic background is important. And most important, I think, is also that we have to be honest. I think if we look at the past decades, what we have done with animal research is that it actually gave us relatively poor predictive validity. So that's something that we just observed, that what works in an animal, the cure, a treatment that works in animal model does not translate always, or very often not, to humans. So we need to to address this and to face this and to come with new models and that hopefully are helping to solve those issues. So one of the aspects that really has been a game changer, and since 2006 we now can actually generate stem cells that we derive directly from the patient's somatic cells. So you think about skin, hair, blood, and you can generate stem cells from that. From those stem cells you can then generate any kind of cell type that you want from of course the biggest advantage that the cells that you produce now in vitro have exactly the same genetic background as the patient. So you solve the problem of the a priori knowledge, because in principle you even don't need to know the exact genetic cause of disease, because you start with the material of that person, and you solve also the genetic background, because you have actually in a dish exactly those neurons, for example, with the same genetic code than the patient you start. Plus, in addition, we know that we are all different, so this genetic background is really important, and I will choose some examples for that. So, a few aspects why I think it really matters to look at to have at least models that recapitulate the human physiology, is that brain development is very different. If I would ask you what makes us so special, I'm pretty sure that you all know that the human brain is very large. We have a very, compared to any animal in the kingdom, we have a very large brain. We actually have a lot of neurons. So it's not per se the size, but it's really the amount of neurons that is very different. Plus the development of the human brain is very slow compared to any other animal. And there is some neurobiology behind this. And just this is a slide, and don't have to look in details, but for example, a mouse, the brain development is a few days. In humans, it takes more than 100 days, and also considerably longer than in the macaque or other big apes. And we now know that there is a neurobiology underpinning of this. And this, for example, lays in the specific presence of cell types that are not present in other animals. And those cells are actually responsible for this, for the fact that we have a larger brain. And so that already says something that our brain development is different than any other species. And so we should take that really into consideration. Now, the other reason that I think is really what we have learned is that although we can identify the genetic causes of these diseases, we are very good at this. But what we see is that if you have a certain mutation in gene, this can actually lead to very different clinical phenotypes, right? So having solved the genotype or the genetic cause of disorder does not tell us so much yet about the clinical phenotype. And I want to illustrate this with a very concrete example. And and that's an example of epilepsy. And now, I just want you to imagine that you have a child is born with a mutation in this gene. We know a lot about this gene, and in the first 2 years, that child will have suddenly seizures, and this can be up to 10 seizures a night. You go to the doctor, and they do a genetic test to find the mutation. And at this point, we are not able to say what the outcome will be for that child for the rest of their life. There are two very different outcomes possible. Either this child will have just febrile seizures, it will grow out of it, and after 2-3 years, it's a normal child. But with exactly the same mutation, that child can develop Dravet syndrome. And Dravet syndrome, that means that the child will keep on having those seizures up to 10, 20 a night, and therefore will have cognitive decline. And at this moment, we are not able, nobody is able to predict what the outcome will be. Yet, the mutation is exactly the same. And the idea behind this, why this happens, is that there is actually genetic background plays a very important role here. So this just illustrates how difficult it is to to actually try to understand those disorders and think about doing this in a mouse model. This is actually not possible because you can mimic this mutation, but then you will just have one clinical phenotype. Now, that's one example, but in the same gene, it's not just one mutation, but we now know more than 1,500 different mutations. So try to also already make 1,500 different mouse models for this, right? So this is, to me, and that is a project that we started, where I thought, well, this is really exactly why the use of stem cells is really beneficial. Will tell us something about this clinical diversity of phenotypic pleiotropy, something we cannot do with animals. So that's one aspect. Now, if we take the same case of epilepsy, then we know that there are quite some anti-epileptic drugs, right? So your child is 2 years old, has a lot of seizures, you go to the doctor. And the doctor has now choice of about 20 anti-epileptica. Now, the difficulty is that at this point, the doctor cannot predict which anti-epileptica really will be beneficial. But what we know is if in 47 or about 50% of the cases, the doctor actually can make quite a good choice. Now, you say that's not bad. But if it fails, the next time you only have 13% chance that the second drug that you prescribe will be successful. And then it dramatically decreases the third time to 4%. And there are different reasons why that is, and I will not go in details. Most importantly is this 36%. Those are the kids that do not have any anti-epileptic drugs that can help them. And so being able to predict that without testing this on the actual patient is really a really important aspect that we need to develop. And so we have to go away from this trial and error on patients, but trying to develop model system where we can actually do this trial and error on a safe platform, in a safe model system. So there again, the stem cells can provide a way out here, because we can really measure the phenotypes on cells with exactly the genetic background of those patients. Now, I think I already explained some of the advantages of using stem cells, and so what you often hear is the word stem cells. I explained what stem cells are, but we use those stem cells actually to generate human organoids. And human organoids are actually nothing else than a miniature version of and simplified version of a human organ. It at least has to contain some of the cell types that are normally present there, but often it's simplified. So it means that you don't find the complicated structures that we have in the brain or kidney, but we find some parts of that. But they are believed, and we think that they really form a very nice experimental bridge between humans and the animal models, with clearly their advantages, which I just outlined before. They are typically generated from either induced pluripotent stem cells, but also from adult stem cells. And I think really where the field is going now is to generate those organs on a chip technology, let's say, so where you can grow them in an in vitro system, where you have full control of all the environment, let's say, and of course you can do that in a very reproducible manner. And I think that nowadays, I think you can make those organoids from any organ that you can think of of a human. So how do we use that now for this SCN1A epilepsy? So that's one of the projects I wanted to just to illustrate very quickly. So we can now start from cohort of patients that either have a control of a very mild form of Dravet or of this Dravet. And we can generate those organoids, brain organoids. And once we have those, we actually have patient-specific brain organoids, and we can measure their activity. And we can measure the activity that is produced by those same neurons. And mind, this is really patient-specific, right? So what we like to do is then actually produce a patient-specific network. And we can do this in high throughput, actually. We can compare a patient-specific network to a control network. And then we can use this anti-epileptic drug and test which one is actually beneficial for which patients. And this is what we want to do prior you would actually do this in a patient. And so currently we are doing this, and we are actually, and I think this is really important, we are doing a retrospective study, right, where we actually doing this on material from patients from which we actually already know which drug worked and which not, to validate the model. Because it's in theory we can do this, but nobody has actually really validated this. So very important. Now there are many challenges that come also along with the use of organoids, and I think you did already mention that in this graph, like green and red boxes, pros and cons. And so I will not go into details, but I think, as I mentioned, the way I look at it is like this here. So you have the sports car is the human brain, and here you can recognize it's a sports car, right? But you also see that it's not driving yet, right? So all the elements are there, and we can study the separate elements, but you can't really you can't drive with it, right? And I like to do that analogy because you did mention that very clearly. If you want to study behavior and cognition, you actually you need a driving car, right? There's no other way. And so that's a really big limitation, that we are able to look at the molecular processes that are happening, but ultimately we cannot link that yet to cognition or behavior. That's just not the system that it's meant for. And that, I think, is really important to understand, that if you have questions about the behavior and the cognition, there is no way you can do that with an in vitro system. Further, there are some limitations regarding the complexity of those models. There are blood vessels in the brain that are missing and so on. But this slide, I made it, I think, now 1 year old, but it's amazing also how, it's moving forward, the field, right? And I think we have to update the paper already. You did, because actually, if I look at it, those 3 points here are already almost solved. We can actually generate blood vessels now. We can link different organoids to each other, that you almost make a mini human in a way. You can link the gut to the brain and so on. And so there is inter-organ communication. And the lack of sensory inputs, well, don't be scared when you see the next figure, but this is the organoids that you can form now. And so this is an organoid, and you can guess what those 2 black balls are here. This is just the retina. We can now generate organoids that typically have always 2 retinas, like you have 2 eyes, and the activity within the brain organoid is depending on light. This is and to me this is amazing, also because we only can generate organoids since 2013, let's say, and we now, even not 10 years later, we can already generate those kind of organoids. And so you can imagine, with the investment that one will do, how it will be in the next 10 years. So those are very legitimate challenges, but they can be solved. In my opinion, the most difficult, and which will never be solved, is of course really looking at behavior, as I explained. So the way I look at it, myself working with animal models and with stem cells, is that this is really it's all about complementary model system, and they are not competing with each other in any way. Even more important, I think that if you want to look at treatments and you want to solve a mechanism, you actually want to have confirmation from different models from different angles. And that's actually, I think, where the debate should be about. And so I participated in this Kdbs report, and a few points that I thought were really important. Again, personally, I think the focus should not be on the animal testing, but rather on the scientific question you want to answer. That should be leading. And then the methods that can answer that question, that's the better, the best. That's the method you should use, I think. And I think also what we are trying to do now is to always find a one-to-one alternative, right? Like, okay, what is the alternative for this, what you do in animal model? How can you find a one-to-one alternative? I don't think that's always per se needed. yeah, and so with this I would like to end, if I can, it's 3 minutes, I think, the perspective of the patient.
Hello, my name is Ashley Point. I'm the president of the Coolant Syndrome Foundation. My 7-year-old son Davis was diagnosed at 16 months with KDVS through a microarray after respiratory infections, hospitalizations, seizures, hypotonia, and many other medical issues. Receiving this diagnosis changed our world and explained everything all at once. We joined a wonderful community of supportive researchers and families. I stepped into the role as president of the Kdbs Foundation after volunteering for several years. The Coolant Syndrome Foundation was created in 2013 with a mission to educate, increase awareness, and promote research for the support and enrichment of individuals living with Coolant syndrome and their families. KDVS was discovered in 2006 by Drs. Coolant, DeFries, and their team at Radboud University Medical Center. KDVS is caused by a microdeletion in chromosome 17q, 21.31, or a mutation in the cancel-1 gene. Once discovered, international teams came together to study all aspects of KDVS, including cognitive, physical, mental, and speech delays. Our families have joined forces with these researchers and have given records and exams, performed surveys, and provided scan biopsies. This data has created a picture of KDVS for newly diagnosed families and led us on a research path. The 100% volunteer-led Kdbs Foundation is dedicated to pushing forward clinical research of this rare syndrome to accelerate and contribute to new therapeutic options and knowledge. We need to find out how, when, and why KDVS occurs. To that end, researchers are using mice and organoids and groups of cells derived from human tissues to serve as neurologic models for this condition. The Foundation has been directly funding research through fellowship and research grants to aid in the development of creating a therapeutic drug or intervention. We understand that research is a deliberate and painstaking march toward truth, and that it requires both patience and funding. Currently, we are focused on the importance of animal models and the development of brain organoids. Organoids promise greater representation of our tissues when compared to cell lines, but offer reduced complexity when compared to tissue explants or animal models. Our current line of organoids is being used in a drug repurposing research project. We're hoping with the ability to create brain organoids, we will be able to better understand brain mechanisms that will lead to direct therapeutic interventions for KDVS individuals. Animal models are invaluable for biomedical research, especially in the context of rare diseases, which have a very low prevalence and are often complex. Mouse models provide information on rare disease mechanisms and therapeutic strategies that cannot be obtained by using only alternative methods, and greatly contribute to accelerate development of new therapeutic options for rare diseases. Our community is anxious for a therapeutic for our KDVS children. Organoids and mice models will help cure my son's speech delay, end Mirabelle's seizures, and fix Miranda's airway issues. We hope to find not only a treatment for KDVS, but a cure and even a prevention. Our families and our children deserve it.
So this is an example of a patient organization which we work a lot with, and at least from our experience is that they really actually see very well all the kind of the pros and cons and of the different model systems, and they are very desperate. They have rare developmental disorders. The pharmaceutical industry is not interested in them because they're just too rare to make any kind of profit. And you see, I mean, this movie. I mean, I just asked her literally, like, would you be willing to say something about, the research you're supporting? And so she didn't get any input from me. And they're actually enormously educated. It's amazing how they dive into the literature and actually get their own opinion about all this model systems and biology. But they are extremely motivated because they have kids with this. And the general public, of course, does not have this time and does not have this kind of motivation to do that. And they rely on us to give the right or an objective idea on how we do research. So with this, I would like to end it. I probably went a bit over my time, Lisa. But thanks for your attention.